Great. Thank you all for joining us today for the Innovators in Cognitive Neuroscience speaker series. Before we begin, let us please acknowledge the indigenous lands from which we're calling in today. The land I'm calling in from and the surrounding area is marked and named by the people from whom this land was taken, the Massachusetts people. Through recognition of past harms, we can resolve to create more equitable and humane futures. I encourage everyone to look up more about the area in which they're living and the indigenous lands in which they're living. So hi, everyone. My name is Daniel Giannini. On behalf of all the students and postdocs on the organizing committee, I'm happy to welcome you to the Innovators in Cognitive Neuroscience speaker series. Collectively, we represent a consortium of schools committed to highlighting innovative advances in cognitive neuroscience, including Columbia, Dartmouth, Gallaudet, Harvard, MIT, University of Pennsylvania, Princeton, and Yale. We acknowledge that these schools are elite institutes of higher learning. So to de-silo the generation of knowledge, we plan to make these talks publicly available on the website with the consent of the speaker. And so that brings me to today's talk. So today we'll be hearing from neuroscientist and philosopher, Dr. David Barrick. David completed his PhD in philosophy at Duke University, and he has worked as a postdoc at the University of Pennsylvania with Michael Platt and Columbia University with Daniel Saltzman, among others. Given his interdisciplinary work as both a philosopher and a neuroscientist, David's research spans a variety of topics, from the neural mechanisms of foraging decisions in non-human primates, to the conceptual foundations of reasoning, to basic questions on the relationship between cognition and the brain. Today, he'll be presenting his work titled Signatures of Information Foraging During Learning of Complex Environments. His talk today will also be done in support of the American Diabetes Association, and there's a link on this slide here, www.diabetes.org, for if you want to look up more information on the American Diabetes Association. So please join me in welcoming Dr. David Barrick.
Excellent. Thank you so much for that wonderful introduction. It's a pleasure to be here today. Thank you so much to the Innovators in Cognitive Neuroscience talk series for having me on, for the gracious invitation. And thank you everyone for joining me today. Today I'm going to present some of my research on reasoning and learning in complex environments and its relation to foraging. I'll be focusing on behavior, but I also hope to get to some preliminary neural data at the end. Let's see here. Let's see if I can turn on the. Is there a way to? Oh, I think maybe. Can everyone see my cursor? No. Let's see here. This is a thing we forgot to test. Here we are. Okay, very good. So yes, thank you for having me on. A bit about who I am first. I am a philosopher and a neuroscientist, as Daniel said. I study reasoning, the cognitive process where agents gain beliefs by drawing conclusions from evidence or from other beliefs. I approach reasoning through the lens of foraging. In particular, when agents reason, they're foraging for premises or inferences, arguments that will allow them to draw conclusions. I contend this is true of formal, even logical reasoning, but also practical and informal reasoning. A number of projects are underway that investigate this conceptual approach, some of which are listed here on the screen. Besides this focus on principles, I'm also interested in the mechanisms for reasoning. I'm particularly interested in the mechanisms underlying non-linguistic reasoning, and maintain that there will be overlap in those mechanisms with the mechanisms that underlie foraging. The focus today will be on the computations that primates must perform to visuospatially reason about their environments. More specifically, how do humans and non-human primates learn and reason in complex environments? Animals must learn how to navigate and operate in these complex environments. These environments, from the grocery store to culturally sophisticated, intellectually inflected academic conferences, to the mundane foraging or socializing context prevalent amongst non-human primates, such as monkeys, often feature complex situations that have many different combinations of features and offer an array of possible actions. Learning how to make decisions in these environments requires the determination of what combinations of features are possible, what actions are possible, and the causal relations between these. One strategy to learn these spaces is by foraging. Foraging refers to area-restricted searches through spaces for resources under conditions of ignorance. All mobile organisms, from bees to hummingbirds to dolphins to mice to a range of different primates, must forage for resources. These resources can be external ones for which animals search in the environment, like food, sexual encounters, minerals, and so forth. They can also be abstract resources like information or reputation, or internal resources like memories, concepts, or strategies, or, as I argue in my work, inference chains, arguments, and sets of premises for reasoning. Humans and other primates, like monkeys in particular, forage in a wide range of problem spaces. Humans are able to forage efficiently, optimally searching for types of food to eat, such as the Aceh hunters choosing what to hunt, foraging through memory, such as switches between categories of animal during recall, or foraging for information on the internet, such as deciding when to leave a web page to try another during surfing. Foraging provides a set of choice algorithms for operating under ignorance, when the forager does not know precisely what they're searching for, where it might be, or what clues in the environment might indicate where to search. When you don't know where to look, you have to use your general expectations about how things normally go as a guide to making decisions. Learning novel or otherwise unfamiliar complex environments is then a foraging problem. When you are learning unfamiliar spaces, you don't know where to look, so you can't rely on your knowledge to decide when to switch things up or where to go next. Instead, you have to use your expectations, for example, the global average as to how things generally have gone in the past. To decide to switch where you are searching or how you are learning, you should make a change if and when you're doing worse at that moment than that global average. In order to determine if agents are foraging to learn novel environments, we need a framework for understanding what animals must learn. Consider this simple example that illustrates what agents aim to learn about the environment. You're at a crosswalk and your task is to get home from work. At the intersection, you can wait or you can cross. If you cross, you can end up on the north side of the sidewalk, or if you're not careful, you can get hit by a car and potentially end up in the hospital. This scenario can be represented using states, actions, and rewards, and the transitions between states. States are sets of features of the world. In this problem, you start in state S1, standing on the south sidewalk. It's going to be defined by various navigational features, such as the scene that you see before you. Actions are the possible goal-directed movements that the agent can select. Here, you have two possible actions. You can go or you can wait. When an agent takes an action, they transition to a state, even possibly to the same state at a slightly later time. So for example, if you decide to wait, you'll end up back in this state S1 on the south side of the sidewalk. If you go, you can end up on the north side of the sidewalk or in the hospital state. These outcomes are associated with rewards. You can get a very negative reward for ending up in the hospital state, for example. Now consider the patch leaving problem faced by all foraging organisms. You're a monkey grubbing on berries at the berry patch. You want to maximize the amount of energy you gather from your efforts. As you gather berries, the remaining berries are fewer and farther between, and they're of lower quality. You always go for the ripest ones first. At some point, you must decide to leave. But when you leave, you expose yourself to predation, a highly negative outcome. In fact, we might even say the reward here is negative infinity if you should be so unfortunate as to perish. On the other hand, as you search, you have the possibility of landing in a new patch with fresh resources, a potentially very desirable outcome. You then have a new decision problem about whether to accept that patch and start foraging or to reject it in hope of other better patches. The key here is that these states, so central to what animals are learning when they're learning how to get around their world, don't come ready-made in the minds of animals. To learn states, animals must learn to represent combinations of different features. These combinations of features are the states of the environment. For example, consider the following features: color, shape, and taste. States correspond to different combinations of features, such as the combination of yellow, curvy, and sweet features into a delicious banana. Enormous numbers of such states are possible. You can see, for example, on the screen below, the combination of a circular or a round shape with color and sweeter taste corresponds to an orange. When there are many features of the environment and animals don't know what features go together, animals are engaged in a search under ignorance, a context ripe for the application of tools from foraging. How do animals learn these states? Animals could be learning by foraging for rewards like food or liquid, or for information like the distribution of features or their locations. If animals learn using rewards, then they will follow the history of rewarding locations to seek the next reward. So for example, after getting some bananas on this hillside before, an animal might travel up this hill to get bananas again. But sometimes animals seek information and they have to trade off a known reward, such as these delectable bananas here, against gathering information, such as traveling up this hillside to gather a new vantage and potentially find new resources. In order to investigate these different strategies for state learning, we want a task that can separate the influence of reward from information on search behavior, that contains different amounts of information and reward, and that elicits different behaviors on the basis of the different amounts of information. A task based on the board game Battleship can separate these influences. How many people here have played Battleship before? This, of course, works better in a live audience, but as a result of the unfortunate circumstances of the pandemic, I can't see how many are raising their hands. No worries, I will walk through the task. I won't assume familiarity with the board game. And there's some differences from the board game as well. In my task, humans and monkeys play a simplified version of the game. In order to maximize reward, subjects must search for and learn the set of hidden shapes over multiple trials. Monkeys searched for shapes with eye movements, whereas humans used a mouse on a computer. In this task, states are defined as combinations of two types of feature: filled or empty squares at different locations. Those states are the shapes that subjects search for. We used a 5x5 grid with these 5 shapes that you see here on the screen. They're the same across subjects, and they always occur in these locations, but only one per trial. Here is a video of eye movements from a sample trial from a monkey. A trial is a series of choices the subjects made to finish revealing a shape. The monkey first fixates, then chooses a square and gets a hit. This rules out 3 of the shapes. Then they reacquire fixation, got a miss, which ruled out the last one of the last 2 shapes, and then they finish revealing the 8th shape, because this trial is from after the animal had already learned this particular set of shapes. As you can see, the animal is very efficient in revealing the shape. The task and timing is depicted here on the screen. Note that color was random from trial to trial, although during this presentation, the shapes will be coded as a particular color. Reward occurs after a piece of shape is found. That's squirts of juice for the monkeys and points for the humans. And there's only a single shape on each trial. Shapes are tied to specific locations, and shapes can overlap. Let's review the example trial in a little bit more detail. Over here on the left, the numbers correspond to the order of choices that the monkey actually made in that sample trial. So first, the animal chose this tile, then this tile, and so on. At the start of the trial, every shape is possible, and the expected reward map of the squares reflects this. On this map here, warmer colors equal higher expected reward on that choice. So this is the reward map before the trial starts. As the animal makes choices, shapes are ruled out and the expected rewards of the squares change. So after the first choice, only the square and the H remain. This changes the expected reward map, as you see here. After the second choice, only the H remains, and the animal then finishes revealing the H. Clearly, there are better choices and worse ones, and which grid squares are better or worse changes over time, depending on what the animal has revealed so far. These changes in expected reward determine which square is best and give us a handy way to measure the animal's performance. Optimal choices occur when the subject chooses one of the squares with the highest expected reward on that choice. Here's the performance for one monkey. Learning performance is measured by the fraction of optimal choices on each trial. On the x-axis is trial number concatenated across 5 days of runs for this monkey. On the y-axis is proportion of optimal choices during a trial. Optimal here, again, means they chose one of the highest expected reward squares. Black trace is the trial-by-trial performance. The squiggly trace at the bottom of the plot is the mean performance across 1,000 iterations of an algorithm that randomly selects remaining tiles on each trial. As you can see, the animal's performance increases over time, plateauing right around 70 or 75%, indicating shape learning, and vastly outperforms the random chooser. My second monkey was even more adept at this task. Here you see the performance curve for all 42 human subjects combined. The thin gray traces are individual subject performances, and the red trace is the average across subjects. Again, on the x-axis is trial number, and on the y-axis is proportion of optimal choices in a trial. Also, again, you see this random chooser algorithm for comparison. The humans, perhaps unsurprisingly, also learn the task over the course of a single 45-minute session and much faster, about an order of magnitude faster than the monkeys. Okay, so we know now the animals improve their performance and appear to be learning the task. There are 3 questions that I'd like to explore in the remainder of the talk. First, do animals use rewards or information to learn about complex environments? Second, do animals use foraging computations to learn those environments? And finally, third, what circuits in the brain perform those computations? With regard to this third question, I'll say a little bit more later, but I have some preliminary data I'll get to at the end of this talk. Before turning to what motivates animals to learn and how, some quantification of learning on the task is needed. To quantify when animals have learned the shapes, we detected change points in the learning curve for each shape. Here, I show the curve for the H shape for my first monkey to illustrate the method. On the x-axis is the trial number for that shape. And on the y-axis is the number of choices for each trial until they finished uncovering the shape. Keep in mind that the shape identity was interleaved from trial to trial. So what I've done here is I've just extracted all the H trials and put them onto one plot. 2 things to notice about this plot. First, there are changes in the mean number of choices to finish revealing the shape. As you can see, it appears as though there may have been a change point right around right here in this learning curve. Second, there are also changes in the variance in the number of choices used to finish revealing a shape. So you can see at the beginning, there was a lot of variability in the number of choices that the animal required to finish revealing a shape. Intermediate amount of variability later on during learning, and very late in learning, there is very low variability. In order to determine when the shapes are learned, we used a change point detection algorithm to detect these changes in the mean and variance. For example, the change point detected in the mean was at trial 34 for this shape. This algorithm was also run on the cumulative variance in a 5-trial-wide window to detect further refinement of learning the shapes. And for this H shape, what you see on the screen, all these vertical lines, were the identified change points. The last change point defined the end of learning for this shape. To the left of the last detected change point is during learning, and to the right is after learning. The algorithm was run on each shape separately, and the end of learning was set to the last change point across all the shapes. Turning now to what drives learning, animals could be using reward or information to learn the shapes. In these heat maps, more red is more rewarding and more blue is less rewarding. In the beginning, The animal does not know which squares are likely to give rewards, and so the values are all equal at the start. As the animal learns, however, the value map becomes differentiated. Eventually, the value map matches what was shown earlier at trial start. So this is what the animal would learn on the basis of the rewards. Alternatively, the animal could be using information to learn the shapes. At the beginning of learning, the different possible shapes are equally likely and uncertainty is highest. Over the course of learning, the probabilities are refined and uncertainty is reduced. The probability mass collects under those shapes and boards that are actually possible for the shape set. Information is defined in terms of uncertainty, so there is greatest information at the beginning of the learning that reduces over time. Animals can use changes in these probability distributions to compute the values of the squares by looking at the change in entropy over the distribution from revealing what is beneath the tile. In these heat maps, more green is more informative and more orange is less informative. At the start of learning, the central squares are more informative because they can be part of more shapes. The information strategy uses the probabilities of the possible shapes and computes the changes in these probabilities when different squares are chosen. The values of the tiles are computed from those changes. At the end of learning, only those squares that inform subjects about the current trial shape are the ones that possess value. To investigate whether subjects learn shapes using information or reward, choice number in a trial was regressed against expected reward, how much animals or humans expected to earn from choosing a tile, expected information, how much animals or humans expected the entropy of the distribution to change from the potential outcomes they might get, and trial number in a run using a binomial model. In this regression, a positive coefficient indicates that a variable motivated earlier choice of a tile in a trial, whereas a negative coefficient indicated motivation of later choice of a tile in a trial. This regression was run on all trials during learning, that is, prior to the last detected change point across all shapes, as I described earlier. Here you can see on average for human subjects, greater expected reward motivated earlier choice of a tile in a trial than expected rewarding ones. This pattern of relative value was also evident in both monkeys. This initial regression suggests subjects were more driven by information than by reward. Granted that animals were more motivated for information than reward, we would like to know how they search for information. And in particular, did they tend to choose nearby squares and sequences, as one might intuitively think for Battleship? Such a search strategy may be revealed by examining sequences of choices. So for example, subjects might start out by searching in a local area, such as this bottom part of the board, for a number of choices in a row before jumping to a new part of the board. Looking for these sequences could reveal their search strategy. On the right panel here, we have probability of choosing a neighbor after a miss during learning. And on the left panel, the same, but for after a hit. On the x-axis is trial number, and on the y-axis is the choice of choosing a neighboring tile, is the probability of the choice of a neighboring tile. The thick black line is the mean across all human subjects, and the gray shading is plus or minus 1 standard area of the mean. During learning, humans start out searching locally following hits or misses, but quickly learn to jump to a different part of the board away from neighbors after a miss. In contrast, following a hit, they tended to persist in that part of the board, and this plateaus right around 70% probability of choosing a neighboring tile following a hit. This pattern of searching locally until a bad outcome is reminiscent of area-restricted search, as I described earlier, a hallmark of foraging behavior. A second hallmark of foraging behavior is a concern with the average rate of resource intake. Recall that foragers should abandon their area-restricted search when current intake rates drop below the global average. Foragers operate under ignorance, without knowledge of when to give up or where to search. The best they can do in those circumstances is to use their expectations based on how well things generally go. When their current outcomes drop below that expectation, they should explore for new resources. Animals across the spectrum tend to overstay just beyond this average. This sort of finding turns up in a range of tasks, including, for example, monkeys foraging in depleting resource patches. Here are the predictions of the optimal foraging model, that monkeys will leave patches when their current intake drops to the average, are in red for different travel times to a new resource here on the x-axis, and the observed time at a resource on the y-axis, with the average plotted here in blue. As you can see, the blue line is slightly above the red line. Monkeys tend to overstay in patches, leaving after intake rates reach the average. This behavior is also seen in humans foraging on the internet. Here, the amount of information available on average is on the y-axis, and the decision to stay on a web page before leaving is on the x. Just as with foraging for reward, humans tended to leave a website when soon after the information dropped below the global average, which is this horizontal dashed line here. We saw a similar pattern of behavior and sequences of choices on our task. On our task, humans tended to jump to a new part of the board after their information outcomes dropped below the average. So on the y-axis here is the information outcome from a chosen tile. On the x-axis is the choice number before a jump. So this is the outcome from 3 choices before a jump, the outcome from 2 choices before a jump, and this is the outcome from the choice just before the decision to jump to a new part of the board. Recall again that a jump is defined as the choice of a tile that was not a neighbor of the current tile. The blue line here is the average outcome across all choices. As you can see, the information outcome of the choice before subjects jumped was significantly below the average. This is completely remarkable and was completely unexpected when we first started collecting data on this task. It's completely empirically driven by these observations, and it was sort of, for me personally, a jaw-dropping moment when I discovered this. It was equally jaw-dropping when I plotted the same outcomes for the monkeys. So here you see the same plot for each monkey. Monkey 1 is on the left and monkey 2 is on the right. Consistent with the humans, monkeys' decisions could also be described as information foraging. Granted that in humans, drops in recent information outcomes below the average predicted jumps, as well as for the monkeys, a signature of an information foraging algorithm, we next asked if there was a connection between how well subjects foraged for information and their ability to learn the shapes. To answer that question, I developed an empirical measure of sort of foraging ability, a forager score. Here you see a schematic of a leave decision to leave a resource, that matches the very general predictions and past findings of foraging. A forager makes a series of decisions and receives outcomes until they decide to leave a resource. These outcomes are compared to the average outcome across the environment, here in blue. When the outcomes drop below that average, the forager decides to depart the resource. This schema can be used to construct a forager score for each subject's set of choices in a row on our task. Each sequence of 3 neighboring choices was scored out of 5 points based on the information outcomes from choices and subjects' subsequent decisions. A sequence scored 1 point for each outcome above the global average 2 or 3 choices before a decision to jump, and 1 point if the outcome before a decision to jump was below the average. Each sequence also scored 1 point for the outcomes 2 or 3 choices before a decision to jump, falling above the outcome just before the decision to jump. A perfect score was 5 out of 5. This score was calculated for each sequence of 3 choices of neighboring tiles in a row, and then the average was taken for each subject. Subjects' forager scores predicted the speed at which they learned shapes. Here on the x-axis is the forager score, as I described on the previous slide, and on the y-axis is the last detected change point, as described earlier. A lower forager score predicted an early, a lower forager score predicted an earlier last change point on the run. A higher forager score predicted an earlier last change point on the run. In other words, the better subjects forage, the more they match that pattern, the quicker they learn the shapes. Okay, so as interim conclusion for the behavior, monkeys and humans both learn these complex environments where there's many possible shapes, and they learn them quite quickly, especially the humans. And monkeys and humans seem to strongly prefer informative tiles to rewarding ones, and subjects of both species also visuospatially forage for information about shapes, and better foraging in humans predicted faster learning. But what are the neural mechanisms that support this kind of state learning? So I'm going to shift now in these last few slides to discuss some neural data. Please keep in mind that these are very preliminary data and analyses. Recordings on the first monkey wrapped up just before the pandemic hit, which, as you can imagine, was subsequently very disruptive to everyone. OFC and amygdala simultaneous recordings have been completed in that first animal with hundreds of neurons from each region. But today, I'm only going to present a subset of sorted data from the orbitofrontal cortex with some preliminary analysis. A recent prominent hypothesis is that the orbitofrontal cortex is important for learning the states of tasks. The orbitofrontal cortex is a large ventral area of the brain that you can see here in this gray, dark blue color that has been extensively implicated in value-based decision-making. The region has extensive connections throughout the brain, such as with emotion and somatic processing areas like the amygdala or insular cortex, and decision-related areas such as the ventral lateral frontal cortex, seen here in purple. In addition, the region receives visual and multimodal input from the temporal cortex. OFC projects to numerous regions as well, including anterior cingulate cortex and ventral striatum, important for action selection and learning. So the region is kind of perfectly situated to integrate all these different sorts of features animals might learn about in the external or internal worlds in order to form representations of states of tasks, such as the shapes on the Battleship task. The task state representation hypothesis for the region predicts that during learning of complex environments, representations of task states should form. I sought to test this hypothesis and explore the role of OFC in forming representations of states using the Battleship task. Much like the behavioral version of the task, monkeys searched for shapes over multiple choices and learned the possible shapes over multiple trials. What you see here on the screen is the task sequence with the timing for those who are interested. Again, color is random from trial to trial, and rewards, squirts of juice, were delivered after the animal got a hit on a shape. There was only one shape per trial and per block that could occur at one of two locations in a prominent deviation from the behavioral version. Once the animal's proportion of optimal choices over the past 30 trials crossed 70%, a new shape and two new locations were randomly selected without signaling the monkey. This allowed me to collect multiple learning curves within a single recording session for electrophysiological analysis. In a moment, I will show analyses of neural data from two epochs: the fixation epoch, which happened once at the start of every trial, and the outcome epoch after a hit or a miss was revealed. Before turning to that physiology data, this is a sample day's behavior. On the x-axis is trial number, and on the y-axis, again, is proportion of optimal choices on a trial. The thick traces are the average performance by block. The thick black line is the 70% threshold for changing blocks. Each color is for a different block, which again contained one shape at one of two possible locations. The first two shapes in the session were learned rather quickly. That's the blue, which was the first, and then the orange. Because the shapes are randomly chosen and I have no control over them, it just so happened that the computer chose shapes for the second block that were rather similar to the shapes for the first block, and so the animal was very quickly able to learn the second block's shapes. A third shape took much longer to learn. That's depicted here in gold. And then the session ended before the animal, the monkey, could learn the last shape here in purple. In sum, the monkey was very adept at learning the shapes on this version of the task, and this session was typical of all sessions. Now I'm going to turn to some analysis of neurophysiology data. But the first question is, what signal in the brain should we search for to test the task state representation hypothesis? What is the psychological correlate of the task state representation, and how might we uncover evidence for it in the brain? Recall the earlier plot of belief state over time, which is shown again here. Shape is on the x-axis, and on the y-axis is the probability of each shape at a location. For this particular schematic, you can imagine that the H was the relevant shape, and these were the two possible boards. As the monkey experiences the two boards for the current block, the estimated probability for those two get higher and higher, and so there's less uncertainty in the distribution. Ultimately, the vast majority of the probability mass collects under those two possible boards. Entropy can be calculated for these distributions, with higher entropy indicating more uncertain belief states. We can then correlate that entropy with neural activity recorded from OFC neurons. To find neural correlates of this belief state, I first examined the firing rates of those neurons during the initial fixation epoch. That's that first fixation on a trial, happens once per trial, when there's nothing on the screen and there were no eye movements. I correlated those fire rates in that 1 second epoch with the entropy of the belief state. Here I'll show an example cell whose firing rate in that initial fixation epoch correlated with the entropy of the belief state. On the x-axis, again, is trial number, and on the y-axis, on this red y-axis, is the entropy of the belief state. The black y-axis here is the firing rate of the OFC neuron in hertz. Note that the firing rate is relatively low. This was characteristic of the neurons in my data set. The solid blue vertical lines are the trials where the shape switched. This red trace is the change in entropy of the belief state over the whole run. As you can see, the entropy starts very high and rapidly decreases as the shape is learned. The entropy also resets a few trials after each shape according to a reset parameter that I fit to the behavior. Following that reset, you can see entropy climbs back up to this high value and then descends again as the animal quickly learns a new shape. This black trace is the firing rate of the sample cell. As the certainty about the possible boards grows over time, this cell's firing rate increases. For the aficionados in the audience, I fit a GLM with a log linear link function and Poisson distributed noise to the observed spike counts with a number of nuisance covariants besides the entropy of the belief state, such as the trial number in run and trial number in block. This cell's activity after that regression significantly negatively correlated with the entropy. Of 175 cells in this preliminary data set, the activity of 57, or about 33%, were correlated with belief state entropy. While neurons in OFC track the animal's belief state, which changes are as shapes are learned, those cells also come to signal the features of shapes as they are learned. The same cell, as I showed a moment ago, also differentiates hits from misses in the post-outcome epoch after learning, but not during. On the left is a plot of the firing rate of the cell versus time, separated by hits or misses during learning. And on the right is after learning. The x-axis is time, with the time of tile reveal, whether it was a hit or a miss, at 0. On the y-axis is the firing rate. This cell shows greater modulation after learning in response to miss outcomes than to hit outcomes, a modulation that was not present during learning. Tiles that are hits are parts of shapes, whereas tiles that are misses are not. And so this neuron comes to signal the tiles that are parts of the shape after learning. Across the population, during learning, about 10% of neurons signaled hits or misses, whereas after learning, about 20% did so. In conclusion, orbital frontal cortex neurons track the animal's belief state and come to signal when tiles are parts of shapes over the course of learning. Of course, this was all very preliminary data, so while these results are tantalizing, it's still early days. Next steps include investigating whether or not I can decode the shape or the probability of a hit or miss from the activity across the population of neurons. Looking for changes in correlations between neurons or changes in feature selectivity during learning. And finally, there's a whole amygdala data set that was recorded simultaneously. So I'd like to see what the amygdala neurons are doing and compare them to the analyses for the OFC neurons. In overall conclusion, today I addressed 3 main questions. Do animals use rewards or information to learn about complex environments? Seems as though humans and monkeys prefer information over reward to learn those environments. Secondly, what computations do animals use to learn those environments? Humans and monkeys forage for information to learn those environments. And lastly, what circuits in the brain perform those computations? I presented some preliminary neural data that suggests that the orbital frontal cortex neurons track the animal's belief state and come to signal when tiles are parts of shapes over the course of learning. And with that, I believe maybe even slightly ahead of time or just on time, I'd like to thank all my funding sources and all my collaborators and mentors. And in particular, I'd like to thank Akram Bakor, who's now at the University of Chicago, for assistance with collecting the human data. Thank you very much for having me on today.
So thank you for the talk. We have some questions on Q&A board, and you can always put more Q&As. So first question from the attendees was that is the difference between the information and reward predictor in your analysis, the prior assumed before learning, flat for reward and center bias for information?
That's a good question. The answer to that is yes. That was the prior that I used for those analyses.
Yeah, so next question is from Yaniv, and then he asked, can you explain the negative influence of reward on choice again? What do you make of that?
Sure. So one of the problems with a complex environment that animals have to learn is that the number of samples or choices that each animal makes of each possible option is actually quite low. So it turned out to be a pretty challenging data analysis problem because we had few to no samples in the vast majority of conditions. So what we did is we sort of reclassified the trials just in terms of which choice number in a trial was a particular tile selected. And the negative influence of reward on choice, given that kind of binomial regression, just means that the rewarding trials tended to be selected later in trials. In other words, tiles that had higher expected reward relative to the influence of tiles that had higher expected information were chosen later. So the way that I interpret this negative influence is just that these subjects preferred tiles that they expected to give information in the sense of they chose them earlier in a trial than those tiles that they expected to give them reward.
Thank you. And the next question is from Kat. She asks, could you clarify the distinction between information gathering and reward search? Isn't any information gathered meant to produce an eventual reward?
Good. Yeah, so that's also a very good question. So information gathering and learning the environment, I conceptualize as part of the cognitive process that animals go through in order to maximize their long-term rewards. So the information that I discussed today is really instrumental information. It's information that subjects can use in order to try to learn their environments in order to maximize that sort of long-term reward, the reward in ultimately in the infinite horizon even. So in this case, information is gathered to produce an eventual reward down the line over the long run. But in the short run, they might choose information over reward. And that's the relevant contrast that I was focusing on today. In particular, I was focusing on that contrast during learning, not after learning. If you run the same sorts of regressions after learning, you see that the influence of expected reward is about equal to that of expected information, which is to be expected, of course, because as you recall, both strategies in the long run will end up favoring the same tiles.
Yeah, thank you. The next question is quite long. So this is from Iviness. So as she understood that you defined the reward of uncovering a tile in Battleship based on just one step into the future, but you could also interpret the reward over a longer period, not just one step ahead. So maybe consideration about reward over long periods subsumes information seeking. Would it be just great to hear your thoughts on this?
Right, so this is actually a really nice follow-up from the immediately preceding question. And as I said in response there, I do think that sort of long-term, over the long horizon, that's why you're gathering information locally in the near term, is in order to maximize your long-term reward rates. I will say, so I didn't present, everything I presented today were just sort of empirical findings. They were findings of analyses of the behavior without or with minimal modeling. I have, as one can imagine, a relatively complicated modeling, actually soon to be paper, hopefully, project that at this point have fit over 100 variants of all different kinds of models to try to tease apart and investigate what best explains choice behavior. And it's important, I would say, to keep in mind a relevant distinction between two different types of things. First is, what is the computation or the strategy that the animals are using? And second, what is the currency that they're using? Are they focused on reward outcomes or information outcomes? It's kind of like a currency question. But then there's also this sort of strategy or computation question. How are they taking those outcomes and turning those outcomes into choices? And it turns out that you can use sort of reinforcement learning algorithms that do have terms for discounted future outcomes, as you refer here, to fit to the behavior, but you can use information or reward currencies in those models. And actually the best fit model, which I didn't present today, turns out to be a variant of a reinforcement learning algorithm called a partially observable Markov decision process that uses information instead of reward. In fact, when I look at the reinforcement learning models and I fit those models with a discounted future outcome, as you suggest here, that discounted future outcome has a fit parameter, a gamma parameter, and that gamma parameter actually tends to drop to 0. So as reinforcement learning theory, if I recall correctly, suggests, when you're in an episodic learning environment, oftentimes the discount factor will drop to 0. And it appears that that's the case. In other words, it appears that that long-term discounted returns from future outcomes is not impacting subjects' choices as much as one might initially think.
Thank you. I think that could also be elaborated in this following session. So next question is from Thomas. This is also related with Battleship. So he asked, how do you think information-based foraging might change the first-person foraging task as opposed to the third-person foraging in the Battleship task?
That is a super interesting question. I have not thought about that. So let me speculate a little bit, which is something I always love to do. My intuition, so a first-person foraging task, let's say you're in the real world. I mean, that's just an individual moving through their very complex three-dimensional environment. And you do see foraging in those environments. And I think there's actually some interesting new results from a variety of labs looking at in controlled settings, looking at this behavior in monkeys and who are moving between resource locations in these large enclosures using wireless physiology. Very fascinating work that a variety of labs are working on. My intuition is that in that kind of circumstance, you would see what is generally observed, where animals do kind of engage in an area restricted search, and then when there is a local decrease in their outcomes, actually below the average, they will tend to engage in this sort of large movement, what's called in the literature a levy flight, to a new location. So I think in a first-person foraging task in the real world, I think you would see something similar. Now, if you're talking a first-person virtual foraging task, suppose you have a monkey using a joystick at a computer, I'm not sure. It's a slightly different type of paradigm. My hesitant prediction is that you would see the same sort of behavior. Individuals would search within a patch, and then when they decided or estimated that they had reached, some sort of threshold, then they would decide to move the joystick so they were headed far away from where they were just foraging to try and find a new patch.
Thank you. So we have 2 more questions, but 6 minutes left. So you could probably choose which question you could answer. The first question is, are there neurophysiological or behavioral markers we can observe of saturated learning? And the second question is, would you expect the strategy to change in your task if there is an aversive stimuli consequences of choosing certain tiles? So you can choose to answer in the Q&A session too, if you think that will take longer, but we have like only 5 minutes left.
Okay, let me see how I can do. The first one first. Are there neurophysiological or behavioral markers? So the neurophysiological question, I'm going to have to punt on. It's still early days yet in the physiology, even just sort of like sorting neurons and things like that. I have a huge amount of data that I have to work through. So I'm gonna punt on that one. And then the behavioral marker, I think, is an interesting question. I can't think offhand. I have looked at, as one can imagine, a lot of behavioral analyses, but there is one in particular that I found, which was super interesting. I don't think I have a figure I can put on the screen for it. I didn't plan for that one, but it turns out that if you look at let me see if I can't get back to the foraging finding, the surprising foraging finding, this one. Okay, good. So this was during learning. If you do a similar sort of analysis for after learning, there's actually no relation. Now, what does that mean? That just means that, because this was, sorry, I did the wrong plot. This one. So if you look at the information outcomes are kind of ignored by the humans after learning. So they kind of shift just to harvesting reward. It's not even obvious it's foraging anymore. So I only saw this signature of information foraging during learning and not after learning. It's like after learning, humans kind of were like, wait a minute, I know where everything is. I don't really need to pay attention to information anymore. They kind of make a decision. These are the shapes. So I'm just going to harvest reward. Somewhat curiously, the monkeys continue to show it after the last detected change point. So there's something that the humans are committing to the states that they think they've learned, the shapes that they think they've learned, in a way that the monkeys did not. And that could be for a variety of different reasons, ranging from the fact that, we gave humans instructions about their searching for shapes to anything, to something ranging to differences, evolutionary differences, or differences in the present character of the behavior of these organisms, whereby perhaps monkeys are just more exploratory in comparison to humans. But the end result was that their reward rates actually were decreased relative to the humans. The humans, by abandoning this kind of information foraging after learning, were able to actually increase their reward rates. How'd I do? Are we out of time? 2 minutes? Should I try?
We have 2 minutes. So we have to end on sharp 1 p.m. We just are going to move the room. So it's really up to you.
Let's see, okay, so this last question is, what would the role be of aversive stimuli or consequences of choosing certain tiles? That's an interesting question. It's a little unclear. I guess for the monkeys, I could use like air puffs. For humans, you could use decrements and points, something like that. And the answer to that question is, I would anticipate faster learning for the reward outcomes. By the way, I should add that I didn't see reward foraging, even though I did see information foraging. So example, if you construct the reward forager score for the humans, which is shown here in a second, I'll get to it right here. This is the same sort of plot reward forager score is on the x-axis. And then, but this is not the info forager score, right? This is for reward outcomes. And on the y-axis is change point again. And you don't see this relationship predicted, the learning relationship predicted that I found for information foraging. If you choose to include penalties, maybe you would see it. I'm not sure. It's an interesting question. And it's something that, on top of many other variants, behavioral variants that we're currently planning on running or in the progress of running, that's something that we should definitely explore. I think I did it. I think I did. Yes.
Yes, you made it clear. Okay. I think that's end of the talk. Thank you for making on time. And yeah.
Thank you very much for having me.
And there's a link